Hi guys, welcome to Learn with Gogs. இன்னைக்கு வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி ஒரு ஆட் பார்த்துடலாம். BuyMote ஷாப்பிங் ஆப். இது ஒரு ஆன்லைன் ஷாப்பிங் ஆப். இதில் க்ராசரிஸ் எல்லாம் அவைலபிளாக இருக்குது. மற்ற ஆப்ஸோட கம்பேர் பண்ணும்போது இதில் குவாலிட்டியும் அதிகமாக இருக்கும், அதே மாதிரி டிஸ்கவுண்ட்டும் கிடைக்கும். ஸோ ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோர் ரெண்டுலேயுமே இது அவைலபிளாக இருக்குது. டிஸ்கிரிப்ஷனில் லிங்க்கும் டீட்டெயில்ஸும் இருக்குது. செக் பண்ணிவிட்டு இன்ஸ்டால் பண்ணி பை பண்ணிக்கோங்க. இது ஒரு ரொம்ப யூஸ்ஃபுல்லான ஒரு ஆப். அண்ட் இதில் ஒரு இம்பார்ட்டண்ட் விஷயம் என்னென்னா டோர் ஸ்டெப் டெலிவரியும் அவைலபிளாக இருக்குது. ஸோ மிஸ் பண்ணிடாதீங்க, யூஸ் பண்ணிக்கோங்க. Now we have to see about plasmid DNA isolation. Okay, first, why we have to isolate the plasmid DNA from a bacteria? Because this isolation of plasmid DNA from bacteria is crucial technique in molecular biology and is an essential step in many procedures such as cloning, DNA sequencing, transfection and gene therapy. This plasmid DNA isolation, this plasmid DNA does not carry any genetic informations. It is extra chromosomal DNA alone. It does not carry any genetic information. So we have to see, we will see the cloning, DNA sequencing by isolating this plasmid DNA. Okay, then difference between the chromosomal DNA isolation and plasmid DNA isolation. In previous video I have detailedly told you about the chromosomal DNA isolation. If you want that video, I'll give this in description, go and check that video. Now difference between these two DNAs is, in chromosomal DNA isolation we will study the genetic cause of disease and we will study the genetic information which carries from generation to generation. Like we will able to study the infectious pathogenic bacteria, their genomes. We have to isolate chromosomal DNA. And in plasmid DNA no genetic information is present here, no. So only cloning and sequencing purpose we have to isolate the plasmid DNA. Here the chromosomal DNA is larger in size when compared to plasmid DNA. It is smaller in size. Okay, and the shape of the chromosomal DNA is linear or circular because it is double helix structures, so it may linear or circular. And in plasmid DNA it is small and circular and extra chromosomal DNA. Okay, and it is found in the chromosomal DNA is found in the nuclear region. Inside the nuclear membrane the chromosomal DNA is present, but in the prokaryotes the plasmid DNA is extra chromosomal DNA only present on the cytoplasm because it is not have well developed nucleus. So it is present in cytoplasm. Okay, the procedures are most commonly same for both chromosomal and plasmid DNA isolation. There are few difference only, I'll told you later. Here materials required, TE buffer is we have to make up to 10 ml. The pH should be maintained in 8, and the components of making TE buffers 10 millimolar to HCl and 1 millimolar EDTA. These concentrations calculation I will give in last. If you want, check it in the last of the video. Then E. coli culture, here we have to inoculate in LB broth. We have 24 horse culture inoculated in LB broth. E. coli culture we want. Okay, then here we have alkali lysis solution. Preparation is 1% SDS and 0.1 normality NaOH, and we have to make up to 10 ml. And this phenol, chloroform and isomyl alcohol we have to kept it a mixture of 25:24:1 ratio, so we get 50 ml. Okay, and we have the second solution only except phenol, chloroform and isomyl alcohol only 24:1 25 ml. Okay, and then we need isopropanol, and the main is neutralization solution. The components are 3 molar potassium acetate and glacial acetic acid. We have to make up to 10 ml. That means 1:1 ratio. That means 5 ml potassium acetate in 3 molar concentration and 5 ml glacial acetic acid. Okay, here in this principle I will tell you the all chemicals role in the isolation of plasmid DNA. Okay, first initially alkaline lysis solution components are SDS and NaOH. These both will go and break or denature the DNA, both extra chromosomal, that means plasmid DNA or chromosomal DNA. Okay, and the neutralization solution, this will play the main role in plasmid DNA isolation, because thus alkaline lysis solution, that solution will denature both DNA, so we cannot able to separate, isolate the plasmid DNA. The plasmid DNA is smaller in size and it is soluble in nature. So when we had neutralization solution, that denatured plasmid DNA only renature and becomes stable DNA. Okay, but chromosomal DNA will not renature because it is larger in size and it is insoluble in nature. Here the neutralization solution used to neutralize the previously added chemical, that is alkaline conditioned chemical. We have to neutralize that chemical, that is help to maintain the pH and control normal environment, which is helps to stable the DNA. Okay, and then LB broth is used to culture the inoculate the E. coli, which contains rich nutrients. So instead of nutrient broth we use LB broth here. This contains tryptone, yeast and NaCl, and 16 to 80 hours culture is very correct stage to use in the isolation of plasmid. Okay, and SGS, it is also known as detergent. It can denature or disrupt the bacterial cell membrane. So in alkaline lysis we use this SDS solution. And phenol chloroform isoamyl alcohol solution. Here each three solution have their separate role. Phenol helps to precipitate the protein. Okay, and chloroform, it will separate two layers like aqueous layer and organic layer. Here aqueous layer only we have to take and transfer to a fresh tube and use. And then isoamyl alcohol, it is used to control the SDS, because the SDS is a detergent. It will provide foams during centrifugation, so avoid that foams, this isoamyl alcohol is used. Okay, and sodium acetate, it is used in chromosomal DNA isolation, not here. And TE buffer, tris HCl and EDTA. Here tris HCl is used to maintain pH, and EDTA, it is used to protect the DNA. This EDTA will go and bind the DNA's enzyme present in the DNA, and that will not able to affect the DNA. Okay, DNA enzymes which present inside the cell will go and denature the DNA present in the plasmid DNA present in the cytoplasm. So to avoid that, this EDTA will go and bind with that DNA's enzyme and avoid the denature of plasmid DNA. Okay, and this 70% ethanol will used as precipitate the DNA. Okay, and agarose gel, it will separate different length pieces of DNA, we know that. And TAE, it used to run electrophoresis by carries current and maintain pH of the medium. Finally we have to know that plasmid DNA is isolated or not by using this gel electrophoresis method. Then finally the procedure. First we have to take the fresh overnight culture in Eppendorf, that is 1.5 ml of E. coli culture in new sterilized Eppendorf tube, and with that 1.5 ml of E. coli sample we have to go for centrifuge, maximum speed at 5 minutes. Okay, then discard the supernatant. Our DNA will set as precipitate, so take that pellet. Okay, so supernatant must be discarded, and that pellet we have to add 200 ml of TE buffer. Okay, and then add 200 ml of this alkaline lysis solution and mix. We alkaline lysis solution contain SDS, it will forms a foam, so we have to mix it gently light, invert the Eppendorf tube and upward. So this gentle mixing will not form any foams. Okay, then incubate in ice. incubate that solution in ice for 5 minutes and add 150 ml of neutralization solution. Then we have to add the neutralization solution and that inverted and upward we have to mix that up and off to evenly mix the solution. Then centrifuge maximum speed at 10 minutes and collect the supernatant and transfer to fresh Eppendorf. This supernatant alone we have to transfer into fresh tube, Eppendorf tube, and to that supernatant add 400 ml of phenol chloroform and isomyl alcohol mixture solution. Then centrifuge 5 minutes at maximum speed and collect that supernatant in a new tube because here DNA will present on the supernatant layer. So we have to collect the supernatant and then 400 ml of here avoid phenol and chloroform and isomyl alcohol only alone we have to form 25 ml solution. That solution we have to take 400 ml and add it to the supernatant contained Eppendorf. Then centrifuge at maximum speed for 5 minutes and now we have to see two layers formed in that Eppendorf tube. That is bottom layer is called organic phase and supernatant layer is called aqueous phase. Here aqueous phase is we needed, so transfer the aqueous phase into a new Eppendorf tube and add 560 ml of isopropanol in that aqueous phase containing tube and incubate at minus 20 degrees Celsius for 1 hour or minus 80 degrees Celsius for 30 minutes. After incubation, centrifuge maximum speed for 10 minutes. Okay, after that discard the supernatant, then wash the DNA pellet. We have to wash the DNA pellet to discard the denatured chromosomal DNAs. If any chromosomal DNA is present in that, we have to discard that. So we have to wash with 500 to 800 ml of ice cold 70% ethanol. Okay, then centrifuge for 5 minutes at maximum speed and air dry and then collect the pellet. Okay, here pellet we have collected, then to that pellet add 50 ml of TE buffer and we have to run the agrosteel electrophoresis and we will get result. We done it in our college and we will get result. This is our result. The DNAs will separate from the bacteria that is seen here by band. Okay, here I will give the calculation for this alkaline lysis and neutralization solution. If you want, check it and prepare and do this procedure, you will definitely get the result because we will get the result. That's why. Thank you for watching. If you understand this video, like the video and subscribe to my channel for more videos. Thank you.